Hello, all NEET aspirants. You are listening to podcast Botany for NEET by S. Ramakrishna. In this podcast, molecular basis of inheritance, the subtopic, the properties of genetic code. The first property, there are 64 codons because there are four types of nitrogen bases either in DNA or RNA, and there are 20 types of amino acids in protein. A combination of nitrogen bases code for amino acid, that is 4 to the power of 3, that is 64 codons are generated. 61 code for 20 amino acid. Remaining 3 do not code for any amino acid, simply functions as stop codon, do not code for any amino acid as it terminates the translation process. Second property, the code is degenerate. Some amino acid coded by more than one codon, and the degeneracy related to third nitrogen base in a triplet code. Suppose GGG, code for glycine, GGA, GGU, GGC. So, amino acid having more than one codon, that property related to degeneracy, that related to third nitrogen base. And degeneracy is not uniform. Some amino acid 2, some amino acid 3, some 4, some it is the 6. And there are exceptions to degeneracy. Methionine and tryptophan are exception to degeneracy. Next. It is nearly universal. The code is nearly universal. Bacteria to human, UUU code for phenylalanine only. Exceptions, mitochondrial codon as well as some protozoan codon. And code is non-overlapping. Some starting fixed point, 3 nucleotides read as 1 unit. No nitrogen base of one codon going to form codon with adjacent nitrogen bases. So it is non-overlapping. Suppose there are 15 nitrogen bases, means 5 codon. It code for 5 amino acid. If more than 5 amino acid means the code, it might be overlapped. So code is generally non-overlapping. Next, code is non-ambiguous. That means no confusion, no ambiguity. A code specifies single and similar amino acid. UUU code for phenylalanine. UUC code for phenylalanine. Of course, phenylalanine having more than one codon, but this code specifies single and similar amino acid. The property it is non-ambiguous, no ambiguity. The code is not having any punctuation or comma. The code is read in a contiguous fashion. No letters are wasted between the codons. And stop codon UAA, UAG, UGA are called stop codon, which bring the termination of polypeptide chain. Suppose in a given sequence AUG, UUU, UUC, UUU, UUC. There is no difficulty in predicting amino acid. AUG, code for methionine, UUU, code for phenylalanine, UUC, code for phenylalanine. Suppose, so in this sequence, we come across the dual codon, that is AUG, as well as UUU, UUC, degeneracy property. Suppose in a given sequence, methionine, phenylalanine, phenylalanine, phenylalanine. There is somewhat difficulty in predicting the sequence of nitrogen bases of the codon because some amino acid having more than one codon. So we can deduce the sequence of amino acid with the help of the nitrogen base sequence in case of codon. Next, mutations. The gene and DNA better comprehend through mutation because literal definition of the gene is very difficult in terms of the sequence. That's why an equivalent term given it was histron. So gene and DNA better comprehend through mutation, that is deletion rearrangement. So there is a loss of gene, gain of gene. Loss of gene, loss of function, gain of gene, gain of function. So a classical example to understand this mutation is sickle cell anemia. Suppose in a coding strand, that is 5'3", GAG sequence, the coding strand sequence is similar to mRNA obtained, that is 5'3". So GAG, it code for glutamate, GLU. In GAG, adenine replaced by thymine, what do you call transversion? Because purine replaced by pyrimidine, that is called transversion. If purine replaced by purine or pyrimidine replaced by pyrimidine, it is called transition. What do you call it? It is a point mutation. Occurs at one base pair change. GAG, adenine replaced by thymine. So in case of mRNA, instead of thymine, it is replaced by uracil, GUG. So earlier, GAG, it code for glutamate. As it is replaced by thymine, substituted by thymine. Thymine in case of RNA, it is uracil. So GUG. So GUG code for valine. So glutamate replaced by valine at 6th position. Suppose in normal beta globin chain, normal hemoglobin, the glutamate at 6th position. In sickle cell anemia, at 6th position of this chain, the amino acid it is valine. So it is sickle cell anemia. So the example for point mutation. And to understand the code, it is the triplet code read in a contiguous fashion. Then example given in the text. Ram has red cap. Ram has red cap. 3 letters which are triplet. Suppose if you insert big B ones, BI ones, BIG ones, in between has and red. Insertion of 1 nitrogen base, insertion of 2 nitrogen base, reading frame affected from the point of insertion or deletion. What do you call the mutation? It is insertional frame shift mutation or deletion frame shift mutation. Suppose if you add the big in between has and cap, Ram has red cap. In between has and red, if you insert the big, Ram has big red cap. So reading frame unaffected. All 3 letters read it as one codon. So frame undisturbed in the segment of DNA. In a segment of DNA, frame undisturbed means it is non-frame shift mutation. So addition of 3 nitrogen bases or addition of 1 codon, multiple of codon, deletion of 3 nitrogen bases, deletion of multiple of codon, reading frame unaffected from the point of insertion or deletion of 3 nitrogen bases. So addition or deletion of 1 or 2 nitrogen bases, reading frame affected from the point of insertion or deletion, what do you call frame shift mutation. Addition or deletion of 3 nitrogen bases, reading frame unaffected, non-frame shift mutation. Next. And another example, UGG, that code for tryptophan. In UGG, all the nitrogen bases in case of that particular triplet codon UGG mutated by all types of nitrogen bases UCAG. All nitrogen bases of one codon mutated by all type of nitrogen bases. How many codons are formed? How many stop codons are formed? How many amino acid it codes? Suppose UGG, first letter change. First letter change results in arginine, glycine. Second letter change, that is UG, in second letter it is the G change, that is leucine, serine, stop codon. Third letter change, that is the G, UGG, third letter is the G. Cysteine, stop codon. So, nitrogen base of one codon mutated by all type of nitrogen bases results in 9 codons. In 9 codon, 2 are stop codon. That means it code for 7 amino acid. In this 7 amino acid, the 5 amino acids which are the types of amino acid. That is the one type of question. Next, the tRNA, the adapter RNA. The RNA molecule reads in one language, speaks in other language. It reads in nitrogen base codon language, speaks in amino acid language. So it is the interpreter of genetic code. Right from the beginning of the genetic code, it was so clear to Francis Crick that there has to be some mechanism to read the code uniquely and also to link it to amino acid. But amino acid not having any structural peculiarities to read the code uniquely. So it is inevitable that tRNA in one hand not only read the code and also link to amino acid. Because other rRNA has a structural role and catalytic role. mRNA loaded with genetic information. Amino acid not having structural peculiarities. So it is inevitable that tRNA not only read the code and also link to amino acid. And tRNA primary structure it is linear. Secondary structure is cloverleaf model. Third order structure, three-dimensional structure it is compact inverted L shape. At one end, third end, amino acid is attached. There is other loop that what we call it is anti-codon loop, which are complement to codons of mRNA. Suppose codon in case of mRNA AUG 5'3", in case of tRNA 3'5", UAC. So AUG in case of codon mRNA and UAC in case of anticodon. So what are complementary segments of tRNA, mRNA? Anticodon, codon. mRNA, tRNA, codon, anticodon. So mRNA 5'3", tRNA 3'5", at third end amino acid is attached. At one loop it is anticodon loop. And each amino acid having specific tRNA. There is initiator amino acid tRNA, that is methionine tRNA, whose anticodons are complement to codons of mRNA. UAC is anticodon, AUG is a codon in case of mRNA. So, initiator amino acid tRNA or methionine tRNA. And there are no anticodons in tRNA for stop codon. Stop codons are UAA, UAG, UGA. So UAA, that is AUU, that is anticodon absent. UAG, AUC, that is anticodon absent. UGA, ACG, anticodon absent. But whatever the anticodons absent in tRNA which are present on mRNA and codon dictionary code for the amino acid. So anticodons are absent for particular sequence of, that is stop codon UAA, UAG, UGA. But these anticodon sequences which are present on mRNA code for amino acid. This is the tRNA is adapter RNA. Actually, before adapter RNA, it was present in cytoplasm in a soluble state. It is called siRNA. It is not snRNA, it is sRNA, soluble RNA, tRNA, adapter RNA, interpreter of genetic code.